I would like to take this opportunity to introduce our next awardee. It's Dr. Claude Desplan from NYU in New York. And I'm going to share my screen here briefly so I can show you what this award really ultimately represents for all of us. So this is the Edwin G. Conklin Medal. And this medal was established in 1995 by the Board of Directors of the Society of Developmental Biology in order to recognize, as the charter says, a developmental biologist who has made and is continuing to make extraordinary research contributions to the field and is an excellent mentor who has helped train the next generation of outstanding scientists. So there's a picture of Conklin early in his youth, and you can see the beautiful, detailed, observational-based drawings that he generated at the beginning of the concept of lineage in the field of developmental biology. This is a snail embryo, and you can see that he began to notice that patterns were emerging with regards to the appearances of lineages. Certain cells were produced by specific blastomeres and the like. And it is this type of knowledge, this type of foundation, that I think has allowed individuals like Claude to do the research that he does today. So Claude is actually very interested in understanding at least three things that I know of. I mean, he's probably many more, but these are the ones I'm going to share with you. He really has been fascinated by how developmental processes make stochastic choices. And he's contributed exceedingly large numbers of information and insight to this particular area. And I presume that he's going to tell us a little bit about it today. Another thing that Claude has been able to do with his trainees and students and postdoctoral scientists is to begin to provide us with a mechanistic understanding of how neural diversity is generated during embryogenesis. And more recently, I've been very fascinated to learn that Claude is beginning to look in collaboration with others, Danny Renner, I believe, in trying to understand how aging and rejuvenation occurs in social insects. So let me tell you a little bit about Claude's trajectory, because it is always illustrative of what it takes, to become a scientist, which is not really a linear relationship, and you really never know where you're going to find yourself, 1 year to the next. Claude received his undergraduate degree from the École Normale Supérieure in St. Cloud, France. That was in the early 1970s. And then in 1976, he started his graduate studies at the University of Paris 7, where he actually worked on vitamin D-induced calcium-binding proteins. Just think about that for 1 second, right? Vitamin D-induced calcium-binding proteins to what Claude is known for today. He then, after finishing his PhD, decided to come to the United States to do a postdoctoral training, a stint in the laboratory of Pat O'Farrell at UCSF. And there he began to study homeodomain mechanisms during development. Later on, in 1988, he assumed his first faculty position at Rockefeller University, where he was an assistant investigator of HHMI, as well as an assistant professor. And then climbed up through the ranks, and in 1992 became an associate professor at Rockefeller, as well as an associate investigator of HHMI. And then in 1999, he moved to NYU, where he's now the Silver Professor of the Department of Biology at that distinguished university. So we want to congratulate you today, Claude, for the work that you're doing, the mentoring that you're doing. And I would be remiss to mention that if you had an opportunity to look at the nomination package for Claude, what became the common denominator in all of the letters was the unanimous agreement of the significant contributions that Claude has made both to science and to the training of his students. And with that, Claude, I'm going to give you a virtual plaque here for the medal, the Edwin G. Conklin Medal, which you will be receiving in the mail at some point in the near future. So Claude, thank you very much for being here, and congratulations, and the screen is yours.
Okay, so I'm the last speaker today, and I guess for the main part of this meeting, and I must say that I'm incredibly proud of this achievement and this receiving this award. And again, this award seems to recognize two things. One is actually the contribution of the people who spend their life in my lab doing some science. And again, I'm very proud of all those people who spend their time there. And I'm not sure if I'm an excellent mentor, but I hope that basically their achievement is a proof that they did something good about them. And actually, several of my grandchildren gave talk in the last few days at the SDB meeting. And again, the best thing is to be proud of the kids and also proud of your grandkids. But this award also, this actually is a list of the people. In yellow as a PhD student, and then white as a postdoc who actually have fairly significant jobs around the world and being through the lab. And again, I hope this list will continue for a while. Okay. Now, this medal also recognizes what we have done over the past, actually, Conklin was quite involved in lineage, and it's something which is a great interest to me. I'm a big fervent believer in that the lineage are very important, and therefore this is actually something which is a great value for me. But again, this is not a live achievement award. It's actually something which has to do with what we are doing right now. And so in continuing what I'm doing now, I want to just tell you how we went there. And as Brigitte emphasized, actually, it's not always a straight line to go from where we stand right now. So when I started as a PhD student and as a postdoc, I was mostly a biochemist, and I was interested, as Alejandro says, in terms of the homeodomain and how the homeodomain protein bind to DNA. We showed it was actually a transcription factor binding to DNA. This led us to be interested in actually the specificity of homeodomain protein and to bicoid, which has a very different DNA binding specificity from most of the Hox gene proteins. And therefore, we looked at the early embryo, and this was with Jessica Trisman in the lab. And this we continued in the lab doing some biochemistry, even going to the structure of protein. And here, what you have on the right side, actually, the structure of a homeodomain dimer binding to DNA and recognizing a sequence of DNA. This led us actually to the binding site for the homeodomain protein, this TAT site, which actually is now being used by many people, as especially people who do some evo-devo, because this binding site, this P3 site, is a binding site for the PAC6 protein that Brigitte mentioned earlier, and it's actually a very common regulator of eye development, a very good marker for eyes in almost every species. It's widely used for doing that. And as Alejandro mentioned, we got right interested in the eye through the PAC6 protein, and then I became a real biologist and also a neurobiologist. And indeed, we got involved in a stochasticity because the photoreceptor in the eye of the fruit fly is actually distributed in a stochastic manner. And you can see here a beautiful image representing the expression of green and blue opsin in the eye of the fruit flies. And you can see there is no real order, it's just stochastic. We spent a lot of time on that. And again, yesterday, a very nice talk from a lab from Bob Johnson has actually taken this work, both in flies and in human, to a new dimension. Another aspect of my lab also is that we are interested in not only the process that exists, and if we use a model organism of the fruit flies to be able to understand that, we are also interested in how this mechanism evolved. And this led us to have a kind of a zoo in the lab of different organisms like butterflies, wasps, ants, even now weevil, and try to understand how those organisms have evolved and adjust their visual system, in this case, to the outside world. So again, this is work, is a very slow transition from biochemistry all the way to the depths of the brain. But and what actually we want to tell you today about what we do now in the depths of this brain. Okay, so we're interested in trying to figure out how the brain becomes what it is, which means how it generates so many neurons of so many different cell types, and how those neurons are going to be wired with one another. Of course, the human brain is inaccessible because with these 80 billion neurons, each one connected to 10,000 other neurons, it's almost impossible to figure out how things happen. And we don't even know the number of cell types or neuronal cell types there is in a human brain. So we chose another brain, the brain of the fruit flies. In fact, not only the whole brain, but actually a part of the brain of the flies, which is the optic lobe. So the brain of the flies has about 100,000 neurons, and 2/3 of those neurons are actually dedicated to the visual system, which means the optic lobe, which represents a major part of the fly brain, contains 60,000 neurons. And we have done, I mean, the community has done an incredible amount of work to try to understand how this brain is made of. And we know that, I mean, starting with Ramon y Cajal and the work of many people, Fischbach, and we know actually how many neuron types there are in the brain. And in this optic lobe, there is about 200 neuron types. So it's a very limited number of cell types. And I should remind you that you cannot fly, flies can do it, and they can do acrobatic flying, which means if you are going to do this acrobatic flying, you better have a very performing visual system. And therefore, we are going to basically to understand how a very performing visual system do function and how it's being built. On top of that, not only do we know a lot about the cell types that exist in this brain, we also know the connectome from the work that has been done at Genelia and other places, where we know how each of the neurons in the optic lobe is connected to each other neuron. It means an incredible level of depth in understanding how this brain is being connected. And therefore, we know how many neurons there are. We know how those neurons are connected. The question is that how do we generate those neurons, and how do you allow those neurons to connect to one another? And I'm going to tell you here today how we can do in terms of single-cell genetics to be able to address these kind of questions. So again, this is actually a view of what we know. This is actually a picture from Fischbach. It's actually a reconstruction from Golgi stainings. And you can see all those neurons that form an incredible network of connections. And as a result of that, what we know is that the flies can actually see the world and react even when it's flying at high speed. Okay? So, so here is actually a better picture of the visual system of the flies. And it's actually a very interesting because it's very well organized system. It starts with the retina here, and the retina is made of 800 unit eyes, omatidia, which means what the flies can see is about 800 pixels in space. Not very much, but as you can see, it can do quite a good job with these eyes. So what you have this 800 detector here, which are these omatidia, they can detect 800 pixels, and they're going to project, actually the photoreceptor are themselves, neurons are going to project to the next layer of the brain called the lamina. Lamina is a very simple structure which has a very limited number of neurons, about 5 cell types only. And as the eye is made of 800 omatidia, the lamina is made of 800 cartridge, each one corresponding to one point in space. And again, this one's a beautiful feature of any visual system is organized in a reticulotropic manner, in the sense that each point in space, each image of the world which is projecting onto the eye, does get projected deeper into the lamina and into the medulla, the next layer. What you see in green actually as a photoreceptor coming from the eye, projecting to the lamina. Not all of them project to the lamina, some project to the medulla, and you can see again these 800 columns corresponding to the 800 pixels detected by the eye. And so on, then the image gets processed, and then the information gets processed and then sent to the higher processing center for allowing the fly to react to the outside world. So again, diversity we know, there is about 20 cell types in the eye, many of them photoreceptors. So lamina is incredibly simple, only 5 cell types, which are mostly dedicated to motion detection. Okay? And then the medulla is by far the most complex structure where most of the processing of information occurs before it gets sent to the lobular complex, where motion vision and all type of visual processing is being understood, in the sense that the visual feature, object detection, color vision, polarized light vision, looming, all of this is being processed independently in the higher layers of the lobular complex. So we want to understand how basically these different neurons are being made. And first thing to do is actually to describe those neurons and to understand how those neurons are being made and what they contain. What do they have in their guts? Okay. So let me try to describe a few of those neurons to give you some idea about how they are being made. Okay. So again, this is actually a picture, a neuron which is going to be part of the medulla, one of those 100 cell types that you see in the medulla. And in blue, what you have is actually the cell bodies, okay? And the cell bodies are all grouped together and send processes that are going to connect to the photoreceptors and are going to basically collect information and process it. And again, the beauty of the system is because you have 800 eyes, 800 cartridge, 800 column, you have basically 800 circuits running in parallel to one another, which for the most part are identical to one another. It's a great advantage because now you don't have one cell type for each of each type, but you have actually 800 cells of each cell type. It's going to be a great advantage for what we want to do if we want to understand how the process is being made. Okay, so let's take one of those neurons. Okay, for example, one neuron that will have the cell body here will project here, and the neuron that you see here, I change the color. The red is a photoreceptor, so basically it's close up to this region here. Okay, and you can see that this neurons has a cell body here. It contacts one of the photoreceptor here. It contacts another photoreceptor here, and then it processes information here by talking to other things, and then go higher focus to higher processing center. We call this a unicolumnar projection neuron because we expect there is one of those neurons for each one of the cartridge, for each one of the 800 cartridges that you have, which means you expect to get 800 of those neurons in the brain. So if you want to make an optic loop, you need to make 800 of those neurons, one for each pixels, one for each of the circuits. But of course, each pixel is not processed independently. There is crosstalk among the pixels, and in the other neurons called multicolumnar neurons, they're going to take information from multiple columns, multiple cartridge. And you can see here, this one, for instance, connect to about 10 cartridge. And therefore, you can imagine you will need fewer of those neurons, maybe if they tile, and to be able to process information because of a much larger receptor field. And you can expect maybe to get something like 75 neurons. So we did this characterization, and generally also did a lot of this characterization, and we came out with a list of basically neurons. And here, just a neuron from the medulla, and they come in different flavors. First, you have the projection neurons. They actually take information from the photoreceptor and send them to higher processing center. And you have local interneurons that simply process information, whatever process means. They remain in the context of the medulla. But what I'm interested today is not so much that. What I'm interested in these two categories I just described to you, those unicolumnar neurons and the multicolumnar neurons. Okay? So we have about 30 cell types that correspond to these unicolumnar neurons. That means 30 cell types, each one of them has 800 neurons. And they can be either projection neurons or local interneurons. Now, the multicolumnar neurons are more diverse, and each one of them is actually a variable number of neurons. It goes from 5 to the lowest number to almost 800 to the highest, most frequent of those multicolumnar neurons, which already overlap with each other. And there is actually a fairly large number of those neuron types. There is 75 of those neurons. That means altogether, you have about 100 cell types that comprise of 30 unicolumnar and 70 multicolumnar. So first thing, how are those neurons made? But first, what are those neurons? How can we define them from a molecular point of view? So thanks to the huge number of tools that have been developed in fruit flies for the last 20, 30 years, and tools that we develop, but mostly have been developed at Genelia, we actually have marker Gulf4 lines that can label a single neurons or a single class of neurons in a body. So what we can do, you can actually use some of those drivers one at a time, and you can actually label with GFP this neuron in a single brain, and then you can take many of those brains, dissociate them, fact sort those neurons, and you can have, for example, a few hundred of those TM1 neurons, which are labeled by a specific line that label TM1. Same thing for TM2, TM3, TM4. We did that one by one, very hard work, and we can get actually the transcriptome of about 20 neurons for which we have some very good lines. And then recently at Genelia, they have continued this work and actually done over 15 neurons, which are part of the optic lobe. It's only 50 neurons out of 200 neurons that comprise the whole optic lobe, 100 in the medulla, the rest in the other ganglia. And we wanted to have a much broader picture because if you want to understand how those neurons get connected to one another, you need to have a statistical power to be able to see how different neurons talk to one another. And so at the time, single-cell sequencing came out, and I'm sure you have seen a lot of single-cell sequencing in the last few days. It's an amazing technique, but it's also an amazing technique, especially for us. Why? Because the brain obviously is extremely complicated, is also diverse, and therefore it provides us with a way to get to the neural diversity on the molecular level in a single snapshot. So what you have here is about 110,000 cells which have been single cell sequenced in the adult, and you can see those cells. Actually, so there's a number here. Okay, 110,000 cells. You can group them by clustering into 193 clusters. 116 of them are neurons, the rest is glia. It's almost a full complement of cells that you expect to find in the optic lobe of fruit flies. Okay? And as I mentioned to you, some of those clusters had been identified because they had been sequenced by bulk sequencing. And we actually did a lot of work to try to identify them based on specific markers. And therefore, we can put a name on 65 of 169 clusters. And therefore, now we know what happened in the transcriptome of many of those neurons, and we can try to analyze in more detail how it happened. But you're going to see it's just the beginning of the story. So again, here what you have, and you can see that you have many clusters, and those clusters are of different size, which, if you remember what I told you earlier in the talk, makes a lot of sense, because you have larger clusters, and those larger clusters most likely are going to be those clusters that correspond to neurons that are present in every one of the colons, those unique columnar neurons, and therefore they're going to have 800 in each one of the brain, and therefore you can see there is a lot of clusters which have about the same size, all of them about the same size, and they actually are going to be the same size, contains about 800 of the same cell types, and you can see that those neurons, indeed, if you look at what they are, for those are known, you can see that indeed those unique columnar neurons. And then you have the smaller clusters, and the smaller clusters are harder to identify because we didn't have any good marker for them, and they always correspond to multi-columnar neurons whose abundance is much smaller. Okay? So this was very nice, and it tells you exactly how the neurons are functioning in the adult because you get the transcriptome of those things. But then we realized actually something that was a bit surprising, and what you see here is that you have this big cluster of neurons. And usually in single cell sequencing, when you get a big cluster of neurons, you say it's crap stuff. It's basically everything else that couldn't be fitting into one specific cluster. This is not the case here, because in fact, this cluster is the best known neuron in the flies. So the T4 and T5 neuron is actually 8 cell type T4A, B, C, and D, T5A, B, C, and D. Those are neurons which are involved in motion vision. Very important neurons. And they all seem to be part of one big cluster that you cannot distinguish, at least in the adult. Let me tell you a bit more about those neurons. Okay, so those neurons, So those neurons actually are neurons called T4 and T5, and they're fairly downstream neurons that downstream of the motion detector are going to receive information to tell them how the fly is moving in which direction. Again, the fly has a retina. The photoreceptor projects the lamina. The lamina, I told you, is involved in the first step in motion processing. It just detects the light, and the motion processor itself, the elementary motion detector, is the set of medulla neurons here. They compare the output of neighboring ommatidia, signal coming from neighboring ommatidia, and can detect the motion, which is going to transfer to the T4 and T5 neurons. Why do we have T4 and T5? Because T4 are involved in the detection of the moving light edge on a dark background. If you have an object, for example, a bird flying, what you can see first, a dark spot moving on a bright background, and then a bright spot moving on a bright dark background. The T4 neuron detects this light edge, whereby the T5 neuron detects a dark edge moving on light background. So this will be the front of the bird, this will be the back of the bird. Okay? And of course, for a fly, it's very important to be able to see the bird. Those T4 and T5 neurons, they come in 4 flavors for the following reason. Because some of the T4A neurons are going to be able to detect front to back motion. When the flies move forward, it basically has the world goes around it in a front to back motion. And the connection in this motion detector here will send information to the first layer of the lobular plate here, where the T4A neuron and T5A neurons are going to project. When the flight rotates, the right eye sees, the left eye sees front to back, the left eye sees back to front, and therefore there is another set of T4 and T5 neurons that detect back to front motion, and they project to the second layer of the lobular plate, and then you have up and down motion, which because of flight is in 3 dimensions, they actually detect those motions in this direction. And again, they project to different layers of the optic lobe. So what's surprising here is that we have very different neurons, either detecting a bright spot, bright edge or dark edge, and they go to project to either the depth of the medulla for the T4, the upper layers of the lobula for the T5, and to different layers of the lobular plate. And yet, in the adult, they're exactly the same. You cannot distinguish them very easily. So we thought about that. We say it makes a lot of sense in retrospect because indeed those neurons do function the same way. They detect, they compare 2 points in space, and they're going to basically compare those points in space. And what matters to distinguish them is not so much how they do it, but how they're connected. Which neurons are they connected to be able to compare either front to back to front, up and down. And therefore, we need to figure out when those neurons get connected, and then you're going to be able to see whether indeed they have differential transcriptome. And so, of course, this happens much earlier in development, not in the adult. So we decided to try to do the same thing, but go back in development and try to see what happens, for example, around 40–50% pupation when those neurons are finding their target, their layers in the lobular plate, in the medulla, or in the lobula. But of course, this is very hard to do because at this stage, the neurons have not yet a form or shape, and therefore it will be very hard to identify the clusters that you can identify here to know what they are. But we thought about the following idea. Maybe what we can do, we can just take our information from the adult and try to match it with fairly just earlier time, like 70% pupation, when the neurons have already acquired, they're just maturing, and then try to match those to the adult, and then from 70 to 50, to 50 to 40, 50 to 30, 30 to 15. And so we just basically sequence those different time points. We made clustering, and then we try to assign a name on all one of those clusters. And this worked beautifully well, thanks to a program developed by Neshet Ozel, a postdoc in the lab, where it did basically it did a transfer of annotation. When you can see here all the names of the neuron we know in the adult, we could transfer them to 70% pupation, then to 50, and then to 40, to 30, and 50. And now what we have actually works absolutely perfectly well, which means every neuron in the adult could have a match in those different time points, which means we can trace now the developmental trajectory of all those neurons from P15 all the way to the adult, including at the time when they basically get made and the time when they're going to project to their different layers and acquire their specificity. And so now if you look at what happened at P50 at the time when they found the layer, now you can see beautifully that the T4 neuron and the T5 neuron indeed group into, actually no longer group into one big cluster. They made 8 beautiful clusters that correspond to those clusters that because they're distinct, because it's when they actually become different to one another. And then later in the adult, they begin to function the exact same way. Indeed, if you look at the distinction between those different neurons, actually the distinction corresponds quite well to the lineage which we have defined. And you can see that actually the front to back and back to front are quite different from the up and down. You can see many different genes are different. And again, you can distinguish between front to back from back to front and so on, and even from the bright edge or dark edge detection. And the beauty of the system, you can identify genes which are specifically expressed in one cell type and not in the other, and be able to figure out how those genes, which for many cases actually are cell adhesion molecule, are going to lead to the projection and the specific connection. And so this led us actually to figure out actually now what we have, all the transcriptome, all the cell adhesion molecule which are expressed in every pair of neurons, and we know exactly which neurons are connected to one another. And the idea would be to try to find which pair of cell adhesion molecule are going to connect them. It's not so easy because almost never do we have single pair of neurons leading to the connection, but through combination and hopefully through some other process, we're going to be able to figure out how basically the system is built to be able to allow those connections to happen. So again, we told you we could match every adult neuron to the earlier developmental trajectories. We can actually, not only in the case of the T4, T5, split the adult cluster into subcluster, but in other type, we could actually see clusters they could split. There were one cluster in the adult, and now they become 2 clusters in P50. Let me give you another example. So DM3 neurons is 1 cluster in the adult. If you look in the adult, you will likely that they become 2 clusters at P50. And why? Because again, they function the same way in the adult, but half of them project in one direction in space, the other one project orthogonal to one another, and they have, again, very well-defined cell adhesion molecules that allows them to project one direction or the other direction. Interestingly enough, some of those molecules which are involved in these orthogonal projections are also involved in a distinction between the T4 and T5 neuron. They're going up and down, or maybe the vertical system and the horizontal system, front to back, back to front, or up and down, down. So again, we have here a huge amount of information that we have to exploit to be able to understand how the process. And we hope that through the connectome and all this information, we might be one day able to figure out what's going on. But again, it's not going to be through each pair one by one. We need to get basically a broad picture to get how all those systems are working together. But then we find something else which was a bit surprising. We found neurons that exist during development, but are no longer present in the adult. And for example, you have these two clusters here. They exist at P30, they exist at P70, and then in the adult, they're no longer there. We cannot find them, okay? And at P30, you can see that a fairly large number of neurons that don't penetrate inside the optic lobe, it's just surround the optic lobe, and then in the adult, we don't find them. So again, at P30, you can see them. At P50-85, later, you can see those neurons. The reason we don't find them in the adult, because they start expressing cascade. They start dying, and in the adult, they're gone. And they're no longer there. To prevent cell death from occurring, those neurons come back in life, and now you have them. Unfortunately, we don't know what those neurons are doing. For those who know the vertebrate cortex, you might recall the chiral rescue cells, which are present in layer 1 and disappear in the adult. And maybe there is something like that involved in the patterning of the brain, which is actually something which is possible due to their localization on the flanking side of the brain. Okay, so what I told you today is that we have a large number of information. We sit on a pile of information, too much almost, and we need to exploit that. We are dealing now, trying to exploit that both from a very broad point of view or from the local point of view as well. But what I need to tell you is that we have all this diversity of neurons. We know how they differ from one another, but how are they generated? How do those neurons get diversified? And how do they come from a neural stem cell to become this 100 of cell types that we get here? Okay? So I'm going to focus my attention on the medulla. And again, the medulla, remember, is made of 100 cell types. And the question is, how do those 100 cell types get made? Okay? So the medulla is made from a single neuroepithelium. And this single neuroepithelium is actually going to be transformed into neurostem cell called neuroblast in flies. And the neuroepithelium is actually a crescent shape here. And at the edge, outer edge of the neuroepithelium, you start transforming the neuroepithelium cell into neuroblast. And what's going to happen actually is that, as you can see here in this schematic, the neuroepithelium is here, and then neuroblasts form at the outer edge. And as time goes, you're going to have the transformation of the whole neuroepithelium into neurostem cells, which means over a period of 3 days, you're going to transform all the neuroepithelium into neuroblasts. And we believe there is about 800 neuroblasts which are being formed there. So this is not actually a random number, because 800 neuroblasts is approximately the number of omatidia, the number of pixels, the number of circuits run in parallel into the circuit. So this was suggestive that maybe what happened that each one of the neuroblasts is able to produce all of the cell types, which means 100 cell types, that surround each one of the unique cartridge. But of course, it's kind of a difficult situation to imagine, because how can one neuroblast produce 100 different types of neurons? In fact, it's almost what's going on there. So question is that how does this happen? Okay, so you make a cross section and you look at the neuroepithelium. As I was telling you, the neuroepithelium becomes a neuroblast. Neuroblasts divide as stem cells, which means they produce an intermediate precursor cell called a GMC that divide once more to produce 2 neurons. And then the next neuroepithelium will become a neuroblast, and so on and so on. And of course, the neuroblasts keep dividing, keep dividing, keep dividing. And what you have at the end, you have all neuroblasts that produce a lot of neurons, and you have young neuroblasts that are just starting to produce neurons, which means you have like a youngest neuroblast, all neuroblasts, and you have young neurons and all neurons here. And so, again, I want to make a very long story short, but we know from the work of Chris Doe and others that actually neuroblasts can actually undergo a transformation. They can actually go through what we call a temporal series, whereby they start producing earlier in their life some neurons, and later in their life they can produce other neurons. And what we have shown actually that is exactly what happened in the context of the optic lobe, that young neuroblasts express a transcription factor called homothorax and produce neurons that express homothorax. And then they're going to age, switch off homothorax, switch on Klumpfuss, then switch off Klumpfuss, switch on homeobrain, then ILS to prepare Dachyteles. And this all neuroblast, it went through a whole temporal series. When it was young, it expressed homothorax, and Clu, then HBN, and ILS to prepare Dachyteles. And every time produced a certain number of neurons. So remember, each neuroblast is supposed to produce 100 neurons. But this series is a bit about 6 or 7. I'm going to add a few more later on. But this is by far not sufficient to produce a whole diversity of neurons. In fact, what happened that there is overlap between homothorax and Clu and homeobrain, which means there is actually a combination of at least 2 transcription factors, which can expand the number of temporal windows to about a dozen of temporal windows. And therefore, what we believe happened actually is that this temporal series is able to produce about 12 temporal windows and to produce 12 different neurons. And the idea will be that every one of the neuroblasts can go through this temporal series and produce about this number of neurons, which can be a bit short as compared to what we want to define. Okay? So the question is that, can we find more of those temporal transcription factors? Because again, they are a bit weird, not enough of those transcription factors. So it's actually a top view. I'm very happy about this picture of the crescent-shaped neuroepithelium. The neuroepithelium is here. The young neuroblasts express homothorax, ILS, solipere, Dachyt. And if we had more color on the confocal, we could actually have more of those. And you can see very well that there's some overlap between the different transcription factors, which allows to create more temporal windows. So we said, let's try to see whether we can actually look at those neuroblasts and focus on those neuroblasts. That means look in the larva when those neuroblasts are being made and transformed from neuroepithelium and produce neurons, and see whether we can identify the transcription factor that change from earlier to older neuroblasts. And again, thanks to all those new software that allows single-cell sequencing to be analyzed and to get pseudo-time, we are able to do that.
Claude, 2 more minutes, Claude.
Okay, yes, I'm almost done. So what you have here actually is single-cell sequencing of many cells in the larvae, okay? And you can distinguish among those cells, again, the neurons, okay, on the left, and then the GMC, those ganglion, and then here, more interestingly, the neuroblast. And again, this is actually the transcriptome of all of the optic lobe. We're going to focus our attention on those neuroblasts here, because we know those neuroblasts actually are those that are going to form the medulla, those that undergo this temporal series. And if you look here and look at just the software, there's something amazing that comes of it. Because if you look at that, you can see here that actually, again, it's just single cells. It's not at all a pattern. It's not an animal. It's just cells on a diagram, a UMAP diagram. And you can see here that at the bottom, you can see the pseudo-time, homothorax, ILS, and I can switch back again, ILS, solipere, Dachyt, and teles, which means now you can reproduce in those cells. You know exactly how the neuroblast can do this pseudo-time and reconstruct the life of a neuroblast from its earlier use to this older age in teles. And what you can do now is take this and see what happened in between. Do you have other transcription factors that go up and down, and indeed we find them. So again, homothorax is there, and ILS, solipere, Dachyt, those were found by Hardway. And now we can find actually a bunch of other transcription factors that go up and down, that correspond actually to other steps, other intermediate steps in the process of temporal windows that actually allow us to understand how you can discriminate and generate more neurons. I could go ahead and explain to you more how you can generate all these 100 neurons, but I want actually to stop here because my time is off and just thank the people who have done the work. Nikos is actually the one who pioneered the single-cell sequencing with the help of Katarina, who actually was doing this BERT sequencing. And then the work has been taken over by Neshat and Felix, who have done the large-scale systems that I just showed you before. And I wouldn't want to forget all the rest of the lab that are going to do an amazing job, and again, allow me to be here today, including people who are doing development, and people who are doing function, and as Alejandro mentioned, we also do some work on aging in ants. And again, these actually are the trainees that actually have left the lab recently to set up their own lab here. And I will not finish without actually acknowledging the support of all the people who are next to me, and especially in the last 3 months, where we've been together with the family. And I just want, if you look behind me, actually, I don't think you can see behind me. You can see and actually the work of my dear wife, Danielle, who has been supporting me for the last, I wouldn't want to say the number of decades. And she's actually an artist, which keeps my sanity, because when I go home, I don't have to think about science. I do think about science, but also I can think about art. And so I want to thank everyone who has been around me all these years. And I want to thank the SDB for giving this amazing recognition that I'm so happy to have received.